the role of biostatistics in medicine and research, organized and conducted by the PG and Research Department of Statistics, St. Thomas College Autonomous Thrissur, in association with the Internal Quality Assurance Cell, the IQAC of the college. We are extremely happy that we are here, and without any delay, we are moving on to the official start. So for the welcome address, I invite Dr. Sajesh T.A., the head of the Department of Statistics, St. Thomas College Autonomous Thrissur. Sir, welcome. Sir, your mic is on mute.
Thank you. Good evening, all. It is with immense pleasure and pride that I welcome you all to the webinar titled Numbers That Heal, the role of biostatistics in medicine and research, organized with the aim of unraveling the indispensable contributions of biostatistics to the world of healthcare and medical research. We are truly honored to have with us Reverend Dr. Martin K., our respected principal, to inaugurate this webinar, whose vision and leadership continue to inspire excellence across all academic and research endeavors. On behalf of all, I welcome you, Father. We are equally delighted to welcome our distinguished resource person, Mr. Ramkumar Rajendran, Director of Biostatistics at Symbions, a leading name in the global biopharmaceutical industry. With his rich expertise and vast experience, Mr. Ramkumar will be guiding us through the fascinating intersections of numbers, data, and the science of healing. On behalf of all, I welcome you, Ram.
Thank you.
This 3-day webinar, scheduled from April 10 to 12, 2025, is designed to introduce participants to the critical role that biostatistics plays in designing clinical studies, interpreting data, and driving evidence-based decisions in medicine. I extend a warm welcome to all our participants, faculty members, students, researchers, and professionals who have joined us from various institutions. Your enthusiastic participation is what makes this initiative meaningful and impactful. Once again, I welcome all of you to this enlightening event. Thank you.
Thank you, sir. For inaugurating this program and for the inaugural address, I welcome Dr. Martin K.A., the respected principal of St. Thomas College Autonomous Thrissur. Welcome, Father.
A warm good morning to one and all. Good evening to one and all. Sri Ramkumar M. Rajendran, Director and Head of Biostatistics, New Jersey, USA. Dr. Sajesh T.A., the HOD of Department of Statistics of our college. Dear experts, delegates, faculty members, and dear students. The title itself is very consoling. Normally, numbers, for those who are not studied, those who are not experts in statistics and mathematics, for numbers must be, especially their elementary school and all, they are frightening actually. Here the title is Numbers That Heal. Healing itself is a very, very apt term or very desirable term these days when everybody is in distress or troubled by so many problems of the day-to-day life, especially in this most modern world. So numbers used to confuse us, numbers used to frighten us, numbers used to put in dilemma and so on. Here you have come up with a novel topic, numbers that heal. So that itself, the title itself is very enchanting. I'm so happy that statistics can offer such a topic, especially in the field of biostatistics. So this webinar, especially during these holidays, for us it is summer holidays, still the statistics department has taken a decision not to rest. They are even working throughout the summer holidays, and this is a 3-day webinar, and including persons, resource persons from abroad, from New Zealand, from what they call Wellington and USA and all. I'm so happy that this is being done. I take this, first of all, I take this opportunity to congratulate the Department of Statistics, especially the HOD Dr. Sajesh T.A. and other coordinating team members, and hoping that this topic, especially in the field of medicine, these statistics has so many roles to play, especially in the matter of healing, that all human hearts and bodies may be healed, and let us have new interdisciplinary solutions to set things, to settle things and have a constellation. What is sought after for all the humanity all over the world. Wishing you all the best for the same. Let me declare this 3-day webinar on the topic healing numbers being innovated. Thank you. All the best.
Thank you so much, Father. Your words means a lot. Now, it's my privilege to introduce the resource person of today's session. Ram Kumar M. Rajendran is a biostatistician with over 16 years of experience leading cross-functional teams in the design, analysis, and interpretation of clinical trials across all phases. He has supported various therapeutic areas, including neurology, oncology, cardiology, and ophthalmology. He represents the biostatistics function in interactions with institutional review boards, ethics committees, and health agencies. Currently, he serves as the director and head of biostatistics at Symbiance, New Jersey, USA. In this role, he oversees the development and implementation of statistical strategies to support clinical research. Prior to joining Symbiance, Ram Kumar held various positions at several contract research organizations, CROs. He holds a master's degree in statistics and has completed a certification in data science from IIT Madras. So on behalf of the organizing committee and all the participants out there, I welcome you to proceed with the session.
Thank you, Gautami. Good evening, everyone. I would like to thank for this opportunity, respected Principal Reverend Dr. Martin, and for organizing this webinar. I would like to thank Sajesh to meet the young minds and share my experience with them. And I also thank who joined this webinar for this interesting topic, analyzing the early phase clinical trials. 1 minute, let me share my screen. So hope you are able to see my screen now.
Yes, sir. Yes, sir.
So I'll be, since we have a very short time, I'll be proceeding with my presentation, and once the presentation completed, you can ask questions, or if I don't have time, or if you have questions later, then you can reach out me in my personal mail ID so that I can answer you for any questions. Please note down my mail ID. Thank you. So actually, we as biostatisticians, the process of drug development is it is a huge process. It is not like some experiment is done and it is the drug is released for the market. It is not like that. From the development of a drug to the releasing of the drug to the market, it takes at least maybe sometimes around 10 years. Even more than 10 years it will take. So during this process, the first step is discovery. In this discovery, for example, in vitro studies, for example, in artificial organisms or the tissues, they will develop the disease, like whatever. For example, if it is a cancer, they will artificially develop the cancer, and whatever the component of the drug they are trying to use for the healing of that cancer, they will use it in laboratory and try whether it is working, whether the drug is curing or not. So once they found that, once they have the evidence that the drug component is curing the drug, then only they will proceed with the preclinical. We call it as preclinical. So in this preclinical, we don't use human for the experiments. So lab rats, for example, lab rats will be used. So in this phase, in this preclinical phase, so the drug for example, like a target dose. What is a target dose that can be given to human, like maximum target dose that can be given to human? So that will be tested in this preclinical. So in this, once the target dose is decided during the preclinical, or if there is any side effects are noted. So if it is, if there are more serious side effects are that will be noted, or it will be stopped during that period itself. So if there are no major serious adverse events, I mean side effects are there, then we will proceed with the phase 0. So that is where the PK. I'll be explaining what is PK/PD and how the statistical analysis has been done for this analysis. I'll be explaining for that. So I just want to give the overall the drug development process and what is the role of statistics. Then I will move forward with some of the examples, how in the early phase clinical trial, how we are analyzing the data. So that will be explained in my coming slides. So in this early clinical trials, it will be patients. Usually patients will be given, and patients will be experimented with the target dose. So once they found that there are no serious adverse event noted, then they will go with the phase 1. So that in that phase 1, we have to check, right, in the healthy patients, what is the drug is doing to the healthy volunteers. Then only we can go and check for whether it is affecting how much it is affecting the patient. So if there is a confidence that it is not there is no serious side effects, we call it as adverse events. So there is no serious adverse events are noted in the healthy volunteers, then only we will go with the patients. So in this during this phase 1 and B, we call it as phase 1, combined phase 1. So here we decide what is the maximum tolerability dose that can be given to a human. So that will be given, whether it should be given before food or after food, whether there is impact on the renal impairment patients or not. So we are comparing the renal impairment, mild, moderate, or normal impaired patients. So we are comparing the drug effect here and decide. Almost it will be decided in the phase 1 itself. Almost we will decide what is the dose of the drug should be given and in what form it should be given, how frequently it should be given. So that will be decided in the phase 1 itself. And phase 2A, again, we'll be checking the safety and the right dose. So there may be some two doses. We might have narrowed down two doses here, and the finalized the dose will be checked in the phase 2A. So once it is done, we will go for efficacy. So there is no serious adverse events, and the right dose is decided. Then in phase 2B, we'll be checking the efficacy. We have to check, right, in large patients. We have to check how the effective of the drug in these patients. That will be checked. And once it is done, we should go for the FDA approval, right? So for that FDA, for example, FDA means it is a regulatory drug regulatory. And in India also we have Indian regulatory. So if there is any drug that has to be approved, it should be approved by a regulatory. Then only it will come to the market. So most of the countries, they follow FDA, Food and Drug Administration, which is in USA. So that and there is a EMA also there. It is a European Medicine Agency for Europe, and even one PMA is that is for Japanese country. So these are some of the leading regulatory department that approves the drug. So for approving them, we need to do the final phase, that is phase 3, where the efficacy and safety information, long-term safety information, all will be collected from the experiment, and it will be submitted to the FDA. So if FDA or any other regulatory department, like as I told, the EMA, PMDA, if we are going to submit to them. So if they are going to validate the results which were submitted by us. So it will take at least 4 to 6 months. Even expedited review, it will take 3 months at least. So that will be for expedited review. We have to pay extra money also. It will be paid by the pharmaceutical companies which developing the drug. So once it is reviewed and it is accepted by FDA, then only it will come to the market. So even after coming to the market, so we'll keep post-marketing experiments will be there. So during the post-marketing only, we will come to know the long-term, though in phase 2, phase 3, we are collecting the safety information, but only post-market will give the exact safety information about the drug. Even some of the drugs will be. We have noticed with serious adverse event after releasing to the market. So that for them it will be withdrawn from the market if it is necessary. So this is the phases of the drug development, and our role of as we statisticians, what we are doing here. So the scientists are developing the drug, and there are medical writers who are experts in writing these results into the CSR, clinical study report, and the FDA is there to approve the results. So what are we doing here? So for example, see, I have from phase from the discovery to the marketing, phase 4, number of studies will be conducted. It will be like maybe 20, 30 also may be conducted, but each study will be designed as a separate study, right? So we'll talk about each study, what we are doing in each study. So we are doing, first there will be a protocol synopsis will be there. So what is a protocol synopsis? It is a short term of the protocol, which includes what is the drug and how it is conducted, what are the statistical analysis will be done. It is a short form of the protocol. Protocol will be detailed. It will explain detailed information about how the trial will be conducted. For example, when is the screening of the patients will be done? What are the inclusion exclusion criteria to include the subjects in the trial, and how they are screened, whether they are getting informed consent. We have to get the consent from them. They are going to participate in this study, so there may be some serious adverse event that will happen to them. So they should agree for that. Then only if they agree, it will be explained to in their mother tongue, whatever the language that the patients understand, or whoever the subject they are entering this study, they should understand what they can expect from this study. So that will be explained to them. They should give the informed consent. Then it will be moved. Then it will be moved, and the assessments. What are the assessments will be collected? For example, how the data will be analyzed and how the drug will be stored. For example, if it is blinded study, how the blinding will be happen, how the blinding will be retained. So all this information will be in the protocol. So in our part, in the protocol development, in our part, we need a sample size, right? How many subjects will be enrolled in this study? How many subjects we need for this study to achieve, for example, 80% of the power. We need to achieve this 80% of the power for the particular effect of the treatment. So for that, we need a sample size. So sample size we have to find out based on the historical information or any other information that is available. For example, if some other pharmaceutical industry, they have done some studies on this particular therapeutic area or the disease. So based on that, it will be the sample size can be calculated, and also of course randomization. We have to do the randomization. For example, in early phase studies, most of them will be blinded, since the healthy volunteers are participating, and the placebo subjects, we call it placebo effect, right? So identical. For example, we are giving a drug, identical to the drug, even color, size, shape, everything it will be given, but it will be no effect, just powder or saline, nothing, no effect will be there. So we have to find the difference, right? Like the subjects who are taking the drug, the subjects who are not taking the drug. We need the comparison. So for that, we need to do the blinding of the subjects. So in this, there are some studies where it will be conducted as an open label. For example, we are comparing two drugs. One is in the form of powder, one is in the form of capsules. We cannot blind them, right? So in that cases, how this will be handled, it will be open label. But as analysis person, study analysis statistician, I'll be blinded until the study completion. So I'll be blinded for it. So that is single blinding. Double blinding, both the patients and who are giving the drug, who are the clinical research associate, CRA. So they also will be blinded, which they don't know which drug they are going to give. And even we will be getting some dry run data sets in between. Interim analysis will be done. So those data, it will be given to us as a blinding only. And analysis method. So how are we going to analyze this data? So this will be us as a statisticians. I'll be giving these inputs to the protocol while writing the protocol. Statistician, CRA, medical writer. So these three department will be combined, and they will be writing the protocol, how the study will be conducted. Then once a study is conducted, the CRF has to be created. So during the CRF creation, also data management will be there, who will be creating the CRF. And as a statistician, we will be reviewing the CRF, how the data is collected, whether it is collected as a numeric or character, how the data, which frequency it is collected, whether they are capturing the data correctly or not. For example, if the inclusion age is between 7 to 18, so while entering the data, it should not accept the 21. So it should accept the data, data between 7 to 18 only. So these will be reviewed by us. For example, data entry guidelines. So how the data entry is done, whether there is any checks required. These will be, as a statistician, we have to do. And database review, how the database is designed, that we have to review. And data transfer guidelines. So how frequently they are going to do the data transfer to us. In what is the structure that they are going to send us in the data. So everything we have to review and give our consent, and it will be approved by us by the digital signature. And during the monitoring also, study monitoring also, we'll be reviewing the protocol deviation. See, we have a protocol, right? How this study should be conducted. So it should be followed, right? That is like a Bible for conducting the trial. It should be followed in the same exactly what is instructed in the protocol. So if something is deviated, for example, a particular drug should not be taken during this during the trial. Sorry. So if that is taken, that is deviation from the protocol. Or if a particular assessment is done, assessment is not done, then it is the protocol deviation. So all these protocol deviations will be collected, and it will be reviewed by us, whether it should be a major protocol deviation or minor protocol deviation. So we will be part of that discussion. And during the data analysis, of course, we'll be writing the statistical analysis plan. So once the data is collected, before the database log, we have to finalize our analysis plan, how the data is going to be analyzed. So there are different populations are there. I don't know. See, I limited my presentation to the phase 1. So there are many things that I don't know. I'm giving. So I don't know. I'm giving whether too much information or that is not sufficient. I'm not sure, but I'm just giving whatever the data, whatever we are doing. Maybe at least you can learn the terms and you can search, Google it, or if you have any questions, you can come back to me. So this statistical analysis plan, there will be population flags will be there, ITT population, intent to treat population. We have to define that, how it will be, what is a safety population, what is the efficacy population that is going to be used. So all this will be defined in this statistical analysis. How are we going to do the analysis? And for example, if you are doing a mixed model, so we have to give, for example, we are using SAS software. So that SAS software example code has to be given to the programmer so that they can follow the code, and because they don't have statistical knowledge, right? So we have to give the model, how the model should be used, and with the example program, so that they can copy paste in their code and they can do the analysis and give us the report. So that will be done. And statistical analysis, even I have to do. For example, even though programmers are doing, I have to do from the raw data sets to statistical analysis. What is the result, so that I can compare with their results. So that will be done, and specifications will be done. Actually, you don't have to worry about that. That is, we have a standard in clinical. We have to, whenever we are submitting the data to the, for example, FDA, we have to follow the standard study data tabulation model. For example, we are collecting the data, right? For example, I can give the variable name as sex. Someone will give us as gender. So there, universally, it has to be followed one single term, right? So that is the standard, SDTM. We call it as SDTM, study data tabulation model. So that the raw data, it may be collected in any form, but that has to be given in a particular structure, so that when we are uploading our data to the FDA or any other regulatory, so that has to be that the data has to follow the SDTM standard. Then only the portal will accept our data. Otherwise, it will not accept. So that has to be converted. The data has to be converted into SDTM. From SDTM, we will be doing the ADAM, analysis data model, so that it is one step away for our analysis. So analysis data model, that ADAM also will be created. And I will be reviewing, as a statistician, I will be reviewing how the derivations are done in the ADAM or SDTM, how it is done. So that I have to review that. And unblinding. Once a study database is logged, we have to unblind. As a statistician, for example, if I am unblinding statisticians, I am the only person will have access to the unblinding, the randomization. So once the study is logged, we have to get the approval form. The study is logged, we can release the randomization. So once I get the approval form, then only I will release the randomizations to the study analysis programmers, so that they can use the actual randomization and do the analysis. And during the CSR preparation, we have to review how the wordings are done, whether it is done correctly, whether the numbers makes correct, whether the numbers make sense to us. So that we have to review the report, clinical study report, that is CSR. So which will be submitted to the FDA along with the data which we have used. So that will be given submitted to the FDA. And regulatory meetings. Once it is submitted, there might be some questions from the FDA. So how this analysis is done, why you have used this model instead of that model, we have to explain them. Why is there any subgroup analysis that can be done? They might ask a few questions. Only for this group, what are the serious adverse events that happened, or is there any particular trend in the adverse events? So that will be asked by the regulatory. So that we have to answer in that meeting. So this is our role of biostatistics in the clinical trials. So in that one, as I told you, it is the phase 1 to phase 4. The process is very long, but I'll be explaining only how some of the statistical analysis, how it is done in the phase 1, early phases, phase 0, phase 1A. Not even preclinical. I'm not going to touch the preclinical. So in the early phase study, as told earlier, so in the early phase study itself, we will be deciding whether it is what is the right dose for the subjects. So even with the right dose, I'll be explaining what is a sentinel subject. Say, for example, we are giving a drug to subjects, right? So while doing the randomization, we have to do two types of randomization within each cohort. For example, cohort means it is a treatment group. For example, we start with the 5 mg drug because it is initial drug, right? So we will go with the lower dose, 5 mg, then 10 mg, then 20 mg, then 30 mg, then 40 mg, 50 mg. So like that, there are 5 groups are there, right? So these we call it as a cohort. So within each cohort, we have to randomize 2 subjects. See, we can directly randomize, but here we call it as sentinel subjects. So that first 2 subjects will be randomized to at least one of the treatment, placebo and the active drug. So that we will have to check, we will have to wait. For example, usually as per standard, within each cohort, 8 subjects will be enrolled. So, but first 2 subjects will be given, first randomized to either one of the drug, placebo or active. So we have to wait and check whether there is any toxicity is there, any serious adverse events are coming. So once the subjects are fine, there are no major adverse events are there, then only we will go with the randomization of 6 subjects. So here in the randomization itself, we have 2 parts. One, we have to do the randomization. It can be first subject placebo or second subject active, or first subject active, second subject placebo. So that we have to randomize, and the rest of the 6 subjects we have to randomize it separately. That may be different combinations, right? Placebo, active, or placebo, something like that. So it can be anything. So the block size also we have to define it properly. Otherwise, the CRA, whoever is working with the patients, they should not predict our treatment, which treatment the next is coming. So that should not predict it. So during the right dose, maximum tolerability dose that can be given to the humans. So that will be identified, and right route, whether it should be given as oral or intravenous or intramuscular or any other route we should use to get the maximum efficacy and safety of this drug, how it can be given. So that will be decided. For example, if you take a paracetamol, if you can give us intravenous, but it will be excreted very fast. It will be excreted from our body very fast. But if you are giving us a tablet, though the dissolve the metabolism done slowly, it stays in our body for the long time. So how long the drug should be in the body, or how fast the rate and the extent. So how we are defining that is based on each drug, and that should be decided during this early phase studies. And frequency, what is the right frequency we should give, whether for example they are giving for kids, first they are starting with the 6 hours or 8 hours interval. Whenever there is a fever, 8 hours interval, we have to give the dose, paracetamol. And what is the dose? That has to be calculated, right? For each, for example, 5 mg per milli, 5 milligram per kg. So that everything, it will be decided during this early phase itself. And the food effect, whether it should be given before food or after food, which one is effective, we have to find out. So that will be done in this early phase trial. And right patient, are we giving the right patient? Whether sometimes the drug will work for only male or female, or particular age group, or some baseline characteristics based on the disease. So our renal group will be there. For example, renal, the renal, right? Kidney functions. So for some patients, the kidney function may be severely it is impacted, or moderate impact will be there. So how these drugs are worked on those patients. For example, if the patients are with diabetes, their renal functions will be sometimes it will be severely impacted. Sometimes it is moderate. How the drug is working for them, and even for hepatic is for liver. How the liver is working for them. For example, some of the patients may have fatty liver, how it is working, or with the normal liver, whether it is working for them correctly. Or so these will be checked during this our early phase itself. So there are number of studies will be conducted in the, this is limited. What I have given is like limited. There are number of phases. For example, drug-drug interaction also will be there. Whether we are giving with another drug, how it is impacting, or if we are giving with another drug, whether it is improving our bioavailability of the drug, whether it is improving or not. So there are many studies will be conducted during this early phase studies. And see some of the studies I have given here. See ADME. How first initially we should whenever there is a drug, we should understand how it is absorbed by the body, how it is distributed in the body, how it is metabolized, how it is excreted. So we have to understand. Here we call it as a first order kinetics. I'll maybe you can search for this term because I may not have much time. Zero order kinetics. So that is like see whenever there is a see how it is for example how it is excreted, right? Usually in the first order kinetics, the Tmax, for example, for example, once we taken the drug, so it goes up to 100 mg in our body. Whenever I take the blood test, it shows that 100 mg in our body. So how much time it is taking to reduce to 50 mg? That is T half-life. We used to call it as T half-life. So how much time it is taking? That is T half-life. So from 50 to 25, how much it is taking? It is another T half-life. From 25 to 12.5, how much time it is taking? That is one T half-life. These T half-lives will be same in the first order kinetics. So whenever there is a first order kinetics, whenever it is proved that the drug follows the first order kinetics, then only we can use for clinical research. Otherwise, if the T half is different for each half, for example, 100 to 50, it is taking some 30 minutes. From 50 to 25, it is taking 10 minutes only. So there is no pattern. It is not constant at all. So we cannot use that drug for clinical research. Mostly they will not use it, and mostly it will lead to failure of the failure of the drug actually in the market. So first thing is here whether the drug follows the first order kinetics or not. So that is important. So that is the reason why the alcohol, the person who was taking his alcohol, it is zero. The alcohol follows the zero order kinetics. So that is the reason the metabolism, the liver metabolizes the alcohol with the constant rate only. It is not like when there is more alcohol, it is metabolism will not happen fastly. It will not happen fast. The drug will metabolize the the liver will metabolize the alcohol at the constant rate only. That is the reason the T of life will not be same. So that is one example. I just wanted to give. And dose proportionality. So that is what. Whenever there is increase in the drug, for example, I am increasing the drug from 10 to 20 mg. It should increase the concentration in our body. So, for example, I am giving 20 mg tablet. When I check after 10 minutes, it is having 15 mg. Then I am doubling my doses. It should double my concentration also after 10 minutes. So that is called a dose proportionality. If there is not dose proportionality, again it is a issue with the drug. We cannot use it for the further analysis. So dose proportionality, checking dose proportionality is one of the very important study we have to conduct in the early phase clinical trial. I'll give explanation. I will give one statistical analysis how we are doing that. We are going to use the power model for that. So let us discuss later in the slides. And obviously, maximum tolerability dose, how it is done. For example, as I told before, so for each cohort will be there, we will be keep increasing it. So whenever there is a grade 3 toxicity is noted, we will stop that drug. So the previous drug will be taken as a maximum tolerability dose. For example, up to 50 mg we have reached, and when it is increased to 100 mg, if there is a toxicity grade above 3 received by the patients means experienced by the patients means, then we will stop at that cohort, and we will consider 50 mg is a maximum tolerability dose by the humans. That we will decide. And steady state, for this steady state, This also I have given an example in the further slides. So the steady state, for example, we are giving multiple dose. So we have to find the interval, right? So when is the say we are giving a drug and there is excretion. For example, you are opening a tab in cup, okay, cup or bucket, whatever it is. So the water is pouring in the bucket. So the at one particular time it is filled, the bucket is filled and it started to overflowing. The incoming and the outgoing, right? The incoming and outgoing, so that is same. So in that state, the water in the bucket remains same, right? So that is called steady state. We think about a drug. For example, each 4 hours I am taking a drug, and there is excretion also. So whenever the intake and the excretion, the intake dose and the excretion is happening, so our whenever this is happening, we have to find out at one point, at one time, the in our body the concentration will be same, right? Like the water level is unchanged, and in our body the drug concentration also same. So that is called steady state. So that is also one of the very important check we have to do for the multiple ascending dose, multiple doses, when we are giving multiple doses. Drug-drug interaction, I have given example for this also. Drug-drug interaction, whether it is improving the bioavailability, whether it is increasing our concentration in our body or not. So that can be given. That can be checked actually. Food effect, whether it should be before or after food, or a hepatic impairment, as I told before, how the liver, based on the liver function, whether it is impacting how the drug concentration impacted by this one. Renal, kidney function, how it is impacting. I'll be explaining what is bioavailability and bioequivalence later. I have given example for this also. So in early phase studies, we will be checking, there are two important things are there. One is pharmacokinetic and another is pharmacodynamics. Pharmacokinetic is how our body reacts to the drug, how our body takes the drug, how it is distributing it, how it is metabolizing it, how it is excreting it. That is talking about pharmacokinetic. And pharmacodynamic, how the drug reacts to the body. What is drug is doing to our body. For example, if it is a cancer study, for example, neutrophil count is there. So how it is impacting the neutrophil count, whether it is increasing or decreasing. So that is how that we are checking, right? So against the time, how it is the effect of the neutrophils, how whether it is increasing or decreasing. For example, I don't know whether you would have heard about fee collection. I mean, for the fee, the patients, they cannot take more protein. So the fee, whenever that has to be increased, the fee has to be decreased in our blood, then only they can take protein. Otherwise, even with a small egg or even quarter of the egg, they cannot take that much protein. They cannot take. So how the fee is increased based on our, whenever we are giving drug. So how the fee is increased, how the fee is decreasing. So we can check the drug. That is called biopharmacodynamics. But we will be focusing more on pharmacokinetic here in our examples. So this is the effective drug. We have to go with a specific disease and we have to discuss. So since I have avoided the pharmacodynamic and we don't have more time for it. So I'm going to concentrate on the pharmacokinetics. So as I told before, this pharmacokinetic involving observe how the body absorbing the drug, how it is distributing to the cells and all, and how it is metabolized in the liver, and how it is excreted. Excreted can be done in many forms. It can be kidney, it can be lungs, it can be even in our sweating. It can be excreted, urine, fecus. So there are many ways of excreting the drug, but most of our drug will be excreted in the fecal and urine. Yeah. So this is what, right? So as I was explaining, say for example, let us take apple pie, right? We are going to eat an apple pie. We are going to cut one, eat an apple pie. So once we swallowed, after chewing it, we are going to swallow it. So it is going to our intestine. From intestine, it will be distributed to the cellular, intracellular, extracellular, other tissues. So it will be distributed that whatever observed the nutrients, right? So it will be distributed to the cells. So from there it goes to the liver where it is metabolized for the nutrients, metabolized. So from the liver it is going to our blood veins. From blood veins it is going to the the nutrients will be going to the blood. So from to the heart. So from the heart it goes to the lung. So where it is oxygenated, our blood is oxygenated, and again come back to the heart and distributed to the artery, so that it goes all over the body. So that is what happening when we are taking a simple snack or fruit. So the same thing it is going to happen when we are taking the drug. So for example, here we are first I'm going to take the drug as an oral, whether it is a tablet or capsule. So it has to go through all the process from intestine to liver to heart, vein, artery, heart to lungs to heart, again heart and artery. But if we need, for example, some of the drugs, it is not given in the form of tablet or doses. So why? Because most of the time it is excreted in the liver itself. So if it is excreted in the liver, the drug concentration is very less entering in our body. So that is the reason some of the drugs are given in the vein. So directly given in the vein, so that it goes to the body, it goes to the heart directly, so that it skips some metabolism here. So during the metabolism, most of the drugs, some of the tablets, most of the drugs will be excreted during the metabolism. To avoid that, some of the drugs are given in the as a intravenous. So once intravenous, this will be skipped, so that at heart, lungs, heart, then artery. That is done. Then again, so for example, immediate effect we need. Some asthma patients are there. We don't have time to get all the drug to complete this process. So directly we are taking as a puff, and so that it is directly goes to the lungs and heart, and it is effectively started working. So that is a different route of administration. I just wanted to give you how the drug is working in our body. And see this one here. So I don't know how many of you identify what is the distribution follows here based on the curve here. I don't know how many of you identify. Let's see. So here, if you see here, when we are giving this one, this purple kind of color. So it is kind of when we are giving in the intravenous, directly it is increased. If you take the blood and check the drug concentration, directly it will be increased. From the starting itself, it will be increased. Then if you see here, the high concentration, it will be excreted fastly. The more concentration in our drug, the more excretion will happen. So that is called first order kinetics. I was explaining. So here, if you see here, next one, intramuscular, if you are giving, it is slowly increasing, and again it is going down. So that is what. If you see up to this one, the more drug, the more excretion. Then when it is going down, the less in drug, the less in excretion is happening. Same goes for subcutaneous, the oral. So if you see here, in this pattern, it is following the log normal distribution. So our analysis will be all the analysis will be done based on the log normal distribution only. So let us first take the dose proportionality. So as I told before, if the drugs are not non-linear, whenever we are increasing our drug, it has to increase our concentration in our body. So if we are increasing our drug, it should increase the concentration. If you are doubling, it should double the concentration in our body. So that is called linearity. 1 minute, sorry. So I think I have already explained the first order kinetics. So in linear system, If it is following the linear, it is following the first order kinetics. So once it is following once we find that it is following the first order kinetics, then only we can go with our drug innovation process. Yeah. So that is what, as I explained before, whenever we are doubling the dose, it should double our area under curve. This one, it should double our area under curve. So that is what should happen, and we are using the power model. Actually, it is alpha and dose power beta. So that is the power model we are using. Since it is following the since as we see before, since it is following our log normal distribution, we are going to logarithmic. We are converting that value. So when you are converting that, it will be log of y. Y means it is AUC, area under curve, the concentration total. So that will be here, and alpha and b. This is the slope. So if we find that if this beta 1 is nearly 1, then we can prove that whenever the dose is double, the concentration also will be doubled. So we can go just a simple regression. We can use a simple regression we can use and find out whether it is working or not. So this is an example I have given here. This is a study design we are using. It is a phase 1 study. It is a single center, double blind. Both the patient and the CRA who is giving the drug, as well as us who is doing the statistical analysis. So all are blinded. We don't know which treatment received by which subject until the study completion, and it is randomized, placebo controlled. Placebo, as I told before, it is nothing. It is not an active. It is a saline. It is nothing. It doesn't have any effect. To check the psychology, sometimes if you see psychologically, we will psychologically I'm thinking that I'm taking a drug, so that psychology itself will cure the disease sometimes. So there is some positive effect to the disease. So that to control that, we are using the placebo control. If it is psychologically, if a disease cured, we don't need a drug, right? So that is placebo controlled and parallel study. That is not a crossover. Mostly bioequivalence we will do crossover study. All other studies will be parallel group. So through the study, they will be receiving same treatment group, and there were 5 cohorts were there, and it is a single ascending dose. SAD means it is not a SAD. It is a single ascending dose study. We are assessing here the pharmacokinetic ADME and tolerability, safety, everything. It will be conducted here. So this is our output will look like. This is area under curve. This is Cmax. I think I didn't explain you what is Cmax. So the maximum concentration here, right? For example, in this case, this is a maximum concentration. At what time it is captured? What is the maximum concentration received in each drug? And under this one, what is the area under curve? So that these are the two. There are many parameters it will be collected, but these 2 parameters are very important for our analysis. Actually, we will be proving. See, once the concentration is collected, see, for example, some of the subject may have vomited the drug. So those will be excluded from the analysis. And some of them, we'll be giving, that is based on this one, we'll be, for example, it has to be given every 2 hours. For example, the drug concentration has to be collected every 2 hours. But instead of 2 hours, if it is collected in 2 hours 30 minutes, so there is a deviation. So if there is more than 20% of the deviation, we will exclude that. 5% of the deviation, we will exclude the drug. We will exclude that assessment or that subject from the analysis. So this will be prepared by us based on the conditions, and it will be given to the PK scientist, pharmacokinetic scientist. So pharmacokinetic scientist will be doing the non-compartmental analysis. There is a tool. There are many tools are there. Pre-vinalin is there. Vinalin tool is there. So based on the tool, they will input this data and they will get out this AUC. We don't have to do manual calculation to calculate the area under curve or Cmax. So it will be given. It will be collected from. It will be derived from that tool. For example, vinalin tool. So that will be done in the vinalin tool, and it will that post vinalin tool we will be getting it, and we will be analyzing the data. So that data only we will use for the analysis. So if you see here, with a simple regression, we have used it here. And as I told before, the slope, if it is nearly 1, then we can, it is unofficial, I'm not informally. We can decide that it is for the drug is following the dose proportionality. But we are going to check the confidence interval. See, as per FDA, if it is between 80 to 120, 20% limit, since it is a log normal, when we are converting it is 0.2. If you go and check the log normal 0.2, if you convert it, it is 80 to 125. It will come 80 to 125. So that is the range we are going to use here. So if it is between 0.8 to 125, then we can call it as a dose proportionality. So if you see here, it is both of them are in the same limit. So here, so since it is the calculated limits are within the range, then we are concluding that the dose proportionality is attained here. So that is what. So we are here intercept, we are not using it anyway, but this is for calculation and how it is impacting. And this is the slope we are going to check here. And the coefficient of determination, how the model explains the variability. So it is nearly, if you see here, nearly it is going to be 100. So it is well explaining the variability in the model. So we can use this model. We can consider this model as effective. So we are done with this check. And next we will go to steady state. So how the steady state, as I told before, it is multiple ascending dose. MAD studies means multiple ascending dose studies. So how within so many intervals, for example, daily once or a day, every 8 hours, every 4 hours, it can be anything. So, but we are going to check before giving drug, the drug concentration will be taken. How in our blood, how much blood is there in our body. So they are called trough concentration. So if you see, they are called trough concentration. So those will be checked. As I told before, like the incoming and the outgoing, which should be same. So then only it is called the steady state. Then only we call it as a steady state attained. So in this method, for each, for example, first point is there. First trough is there. For first trough, for example, in this case, 5 troughs are there. 5 troughs sample collection, day 2, 3, 4, 5, 6 are collected. So in each trough, this day 2 will be compared with the mean of the other days, how it is done. So in that way, if it is different, if it is same, then we can decide that the steady state is attained at day 2 itself, or it can be attained in day 3, or it can be attained day 4, or it can be attained in day 5. So here we are using the Helmholtz contrast, which is you can use anything. Here you can put 4, -1, -1, -1, -1. So obviously it is going to be 0. So that as a statistician, so you know this logic, right? So yeah. So let us see an example. So here, if you see the study design, it is a phase 1, again parallel. It is an open label study. As I told before, for some reasons it is good. It could not be blinded, and it is a MAD study. It is multiple ascending dose study from day 8 to day 17. They are collecting the trough. So we are assessing the PK here. So as I all like previously we checked, right, AUC, Cmax or Tmax. There are many things that the parameters will be there. So all will be summarized, but here I'm focusing only on the steady state. So here, if you see here, from from day 9, the trough day it will be given, and next day it will be collected, the trough concentration will be collected. So here the first step, day 9 to the remaining days, it is matched, the compared. Again, day 10 compared with the remaining days. Day 11 compared with the remaining days. Day 12 compared with the remaining days. Like that it goes on. So day 16 to day 17. If you see here, this one less than 0.05. So there is some significant difference there. So we cannot call that here we have the steady state as attained. But if you see the next one, if you have a null hypothesis, there is no difference between these two groups, right? These two groups. So that means that they are the same, both are same. So in case we call that And if you see here as well as the 80 to 120, it is between these two ranges. So we call it as the steady state is assigned. I mean, steady state is attained at the day 10. So day 10 it is achieved. And bioavailability, I have two more slides only, so I will complete fast. So the rate and the extent of the bioavailability means, see, for example, we are taking a drug, and at what time, what is for example, I'm having a headache. So here the drug, how much drug is available at the place of action, how much is there, how fast it is there. So that is called bioavailability. So how that bioavailability can be checked. For example, we can check non-intravenous and intravenous. We can check. That is called absolute bioavailability. For example, if you are checking non-intravenous between two non-intravenous, then it is called a relative bioavailability. So, and here, if you see here, I'm checking. We are checking drug. See, we are giving one drug against a single drug. We are adding one drug plus, see, we are giving X, and again with another group, we are adding one more drug to the X, so that how the bioavailability, how much, for example, at that, for example, I'm having headache, so how much drug is available at the site of action if I add one more drug to the current drug. So what is the how it is checked? Let us see. So here we are using the mixed model, and the of course log transformed to PK parameters as we are about the log normal with the fixed effects of treatment. Treatment will be a fixed effect and random effect for the subjects who are participated in this. So that will be done. So we have a SAS software proc we called. I mean our model is called. I mean procedure is called proc mixed. We can use that, and by using that we are checking for the AUC and Cmax. These two parameters we are checking, and if you see here, compare this one, it is increasing when we are adding the drug X, drug Y to the X. It is increasing the bioavailability to 74%, and here at least by around 50%. So it is increasing the bioavailability when we are adding another drug to this another component to this drug. So that is a conclusion here, and bioequivalence. See, we are checking whether they are equal. If they are equal, the effect also will be equal. Right? For example, here in India, if you see many drugs will be there for the same compound. Many drugs will be there, so they are called genetic drugs. So usually there will be a pattern will be retained by the pharmaceutical industry for some period, for example 20 years. So once it is expired, it will be out for all other companies. They use the component, so they can use it and reproduce the drug with for lesser price. So that is what happens. So when we don't have to do the same analysis here. For example, if it is done, for example, this drug is done for America, and if the same drug we are going to apply for India, just check the equivalence in the Indian subject. If they are equal, the effect also going to be equal, right? So, so that is what they are doing here in bioequivalence. So here, if you see, here is an example. Here we are using the crossover. As I told, for this study we are using crossover. And of course, the PK parameters will be log transformed, and sequence period treatment, and nested volunteer within 6 months will be fixed effects. So we are using that, and here if you see, we are checking two treatment E and F. We are checking the bioequivalence, whether it is equivalence or not. If you see here, it is not equivalence because it is not within the limit, right? So it is not within the 80 to 125, this limit, confidence limit, if you see, it is not within the limit. So that is the reason it is not meeting the bioequivalence. So for example, here if you see, it should be within the confidence limit, should be within this 80 to 120%. Then only we can call it as bioequivalence. Even at least one limit, if it is outside, we cannot call it as a bioequivalence. It is not meeting it is not included here in these two cases. But if you see here, it is out of the limits. So it's a bio-inequivalence. We can directly call it as bio-inequivalence. So thank you. Thank you again, Dr. Sajesh, for this opportunity. And if you have any questions, please you can ask, or if you have any questions later also, you can mail me the mail ID which I have given.
Participants, if you have any queries, please feel free to ask in the Google Meet chat box or in the YouTube live chat.
Good morning.
Ram, in both the examples, it was that 90% confidence interval was used, is it?
Yes, 90%.
Yeah, we used to take it as 95 or 99, something like that. Why? 10%?
Yes. I mean, I expected this question actually. See, this is 90%. 90% is defined by FDA and EMA. So it is okay to achieve within 90% confidence interval. If it is achieved within 80 to 125, we can call it as bioequivalence. It is already the bioequivalence conditions were released by the FDA and EMA. So we are following the 90%. The alpha level is going to be 10%. Yes.
Okay.
Yeah, 90%. Yes.
There's one question from Ramya Nadeshan. In sentinel subjects, if we are doing randomization of first two subjects, means either they can receive active drug or placebo, or both can receive the drug. This can also happen.
No. That is the reason. Sentinel subject. When we are defining this sentinel subject, the first two subjects will receive either one. Either one of the treatment. It should be received either placebo and active. One subject should receive the placebo. One subject should receive the active. That is the condition for the sentinel subject. So it is not like that is the reason I told the randomization. There is two part. For sentinel subjects, we are going to consider two, right? So for example, active and placebo. So active will be assigned to one subject, placebo will be assigned to another subject. So it can be a first subject or second subject. For the rest of the 6 subjects, there are 6 subjects, right? All the first 3 subjects may receive active, all the last 3 subjects may receive the placebo, or first 2 subjects may receive, it depends on randomization, right? So that can be changed. But for sentinel subject, the condition is one subject should receive the placebo, one should receive the active. But the order may be changed. Either it is placebo or next subject is active, or the first subject is active or second subject is placebo.
Next from Srividya Balasubramaniam. How do biostatisticians determine an appropriate sample size for a clinical trial? And what factors influence this calculation?
Right. So as I mentioned earlier, see, we would have done number of research within before we are coming to here. For example, even sometimes the same for the same disease, some other pharmaceutical might have done the research and they might have released some results. So based on that, we can decide the sample size, or we'll be doing within our pharmaceutical we might have done some research, or if there is any published papers are there, we can use that for the sample size calculation. And impacting, that is what I see. Whenever you are going to increase our power of the sample size, it is going to increase the sample size. So whenever you are going to the variation plays the important role here. So so these are the things, variance, or power calculation. What is the power you decide? What is the effect size? So these are the three things that is going to impact our sample size, whether it is going to increase or decrease.
Okay. Next question is from Professor Dr. Davis Montasheri. Can you suggest any reports already published in journals dealing with this type of different phases of drug development and applied various statistical tools for data analysis crossing various phases?
Yeah, this one I thought of telling you, but I forgot this one. You can go clinicaltrials.gov.in, gov, clinicaltrials.gov. So all in most whatever the studies that is submitted to the FDA, all the results will be published there. For example, even I refer some of the studies from there. For example, I'm working on a new therapeutic area. So I don't know these endpoints, how they are analyzed, whether they are categorical or numerical. There are many therapeutic areas, right? We may not know how each endpoint is designed or collected and analyzed. So I go this clinicaltrials.gov and I search for this. You can search the terms. For example, health-related questionnaires. You can go there. For example, mFOREST is there. I never heard before mFOREST score. So what I do, I go and search in that one, and so there are a number of studies already. They would have done a number of studies how they would have analyzed the data there. So I go and check there how it is analyzed, check there and apply the same, or I can make it better than that analysis. We can suggest, or we can do some published papers based on that. I can apply our statistical analysis.
Is it from US government or?
Yes, it is from US government. FDA, it is free for, it is open to everyone. It is open source. They will give what is the study. They will give the details like what is the study design, how much subjects enrolled, whether there were any adverse events, the count and percentage, or if they have done any statistical analysis, that also will be listed. Even the statistical analysis plan will be included, protocol, everything will be included.
I hope that's all. That's all the questions. Gautami, please.
Yes, sir. So with that, we have come to the end of today's session. For the vote of thanks, I invite Ms. Amrita M., research scholar from the Department of Statistics, St. Thomas College, Autonomous Thrissur.
Thank you, Gautami. Good evening, one and all. Respected Dr. Martin K., our beloved principal, and Dr. Sajesh T.A., Head of the Department of Statistics, St. Thomas College, and today's resource person, Mr. Ramkumar M. Rajendran, Director and Head of Biostatistics, all the participants gathered here, and the faculty members of the department, and my dear friends. Today, we had a very fruitful session on the topic Analyzing Early Phase Clinical Trials by Ramkumar Sir. And on behalf of Department of Statistics, St. Thomas College, and all of us gathered here, I extend my deepest gratitude to Mr. Ramkumar Sir for his expertise and willingness to share knowledge have truly enriched this event. And I hope you guys all get well by this session. And this is very, he introduced pharmaceutical as well as clinical trial terminology to us. And I would like to extend our heartfelt gratitude to our principal for inaugurating our 3-day webinar on the topic Numbers that Heal the Role of Biostatistics in Medicine and Research. Also, I wish to thank our HOD Sadesa for his generous support, without which this webinar would not have been possible. And thank you all the participants present here for your valuable presence, and I hope you all gain something beneficial from this session. Once again, thank you so much.
Thank you.
Thank you, Amrita. For your information, tomorrow we will have our speaker, Harini Srinivasan. Her topic is on the role of biostatistics in modern healthcare. And the day after tomorrow, we have our speaker, VRC. Her topic is on applications of biostatistics in medical research. And you can use the same Google Meet link for joining tomorrow's session. And once again, I thank all the participants for your active participation. See you all tomorrow. Thank you, Ramkumar sir. Have a good night.
Thank you.